Hello, and thank you for being here. Hi, I'm Jen Prince, Miss Prince, and we are going to go through a little presentation to get you ready for a lab where we're going to look at homologous skeletons. We are going to see how the principles of evolution allow us to look at some known specimens, some familiar animals, and after studying known examples, you will be able to identify some unknown bones using these principles of evolution. Yes, so we'll go through this. Hopefully this little screen sharing thing isn't blocking you too much. I'll find out later. Okay, I wanted to start by sharing this little, showing the different ancestors of modern day horses, since this came up in your packet recently, where you're looking at the skulls that have changed over time and the forelimbs, seeing this prehistoric horses having hands. That was exciting to us. So, let's get into it. I'm gonna move this just in case it's blocking you. Evolution is change over time, not within one individual, but across generations. So this here in the lower, should be your lower left, is representing a simplistic version that is showing the interconnectedness and the relatedness of all living things. All life on Earth, be it bacteria, archaea, or things like us, multicellular things with nuclei, our eukaryotes, all have a common ancestor. And that from small genetic errors and random mutations that have accumulated over time, we now have this diversification of life. So here you're seeing some Galapagos finches and then different mammal jaws, showing again their similarities. We'll go into it. They're not just similar, they're homologous. And homo meaning same. There are many different traits that are shared within similarly related species and then more distantly related, as you guys studied with these arm bones. Here, this diagram is going to be the first of a few we're going to look at that'll show we vertebrates and the changes that have occurred to us that make different types of vertebrates. We're going to go into this, but you can see at the top we've got one label of chordates. So this is everything with spinal cords. Not everything with a spinal cord is a vertebrate. So we see these different changes that led to the speciation events, those branching shapes that you see at the bottom here. But just to look at the top again, you can see everything up there is a chordate, but not everything is a vertebrate. Some things have jaws with bones, lots of changes accumulating. We'll look at more of this in a moment. Okay, homologous structures. Homologous, homo meaning same, these are the same structures or similar features in organisms that are descended from a common ancestor. So again, homologous, different from analogous, where analogous would be more like a bat wing and a butterfly wing, have similar shape, have similar function and adaptation for the organisms that they serve, but they are different. They come from a different genetic influence. Sorry, I'm laughing at the bird messing around behind me. But yes, again, homo meaning same here, the same structures, and they have a common ancestor. We are vertebrates, and we are defined as having an endoskeleton. This is part of what lets mammals and other vertebrates grow to such large sizes, is with all that bone and cartilage, we can support a lot more weight than an exoskeleton. So you can see our little crab in the corner here. The chemical structure of those skeletons, there's only so big they can get, and then we have other factors that come into play. Really quickly wanted to share a little bit of, so homo can mean same. It also, that was in Greek, in Latin, it means human. And so we homo sapiens are humans that are thinking, but we have a lot of different, what we would call hominid or different cousins within our part of the family tree here. All of which do not exist anymore, except for the small amounts of their genome that are retained within us, but that's another conversation. And I don't want to leave you out, so I'll share with what I share with the classes. A really exciting one in the middle here is Homo floresiensis. On the island of Flores in Indonesia, is one of the more recent humanoid or hominid discoveries of these naturally dwarfed people. They were 3.5 feet tall when fully grown. If you look up Flores man, Flores, it's pretty exciting that these hominids, these pseudo-humans were coexisting at the same time as early humans, only 120,000 years ago. again, more with our human ancestors and kind of following this theme of homology, that we can see similarities in the bone structure of these different hominids, and where DNA evidence cannot be found or is always able to be taken out of some bones like this, we can use the physical evidence of these shapes of the bones to look at our ancestors and try to put together the puzzle pieces. Here, I won't go too far into this. If you want, there's links to all these articles and quotes that I'm pulling out if you want to do more research over the summer. But in 2 million years ago, as our early human populations were beginning to move out of Africa or within, there was still a lot of gene flow between Europe and Asia and Africa. We have been one fluid population, but it's a big conversation. Okay, these diagrams, I've been showing you a few different types of what we call cladograms. You may also hear them pronounced cladograms. They are these diagrams showing the similarity or relatedness of different organisms. As I stop talking, if you look at these, you can see they are different. I want to share these are both accurate. They're just different ways of classifying these organisms. The one on the left, you can see is labeled as classification based on visible similarity. This is what you might be doing out in the field. We're just based on its shape, how might I relate these? How might I guess at their relatedness to each other? We'd probably say, hey, conical shells, the barnacle and the limpet must be more similarly related. This crab with its appendages, very different. We'll put it over here. We have access to genomes and DNA sequencing that allow us to build much more accurate cladograms and really reevaluate a lot of these relationships as we understand evolution better. So the one on the right is going to be more accurate, where everything after that evolutionary change of developing tiny free swimming larvae, all of the above have that trait. You can see this branching event occurs where only the crab and the barnacle have segmentation or exhibit molted external skeletons, whereas the limpet is more different. It feels okay. What's key to remember is that these are both correct, but there might be one that is considered more accurate for our needs. Beginning a little cladogram bonanza, but I promise I'll keep myself restrained. This is showing vertebrates, things with bones, and showing some of the traits that are shared amongst these creatures. But you can see, looking at the top, we've got sharks, tunas, frogs, humans, representing a couple different groups. Everybody on this list has the first trait that's being shown, where they all have a vertebral column or spinal column. But they do not all have a bony skeleton. Sharks here have a cartilaginous skeleton, so they're out. And again, you see that narrowing of traits. What I want to draw attention to is not everyone that is super related or more closely related will have the same trait. You might have a loss of traits. You may have a trait be lost and then regained. Snakes, for example, come from a reptilian ancestor that had 4 limbs. And so if you didn't know, snakes have pelvises, snakes have hip bones because they came from an ancestor. They have lost their appendages, they have retained their hips and their pelvises. There's only one freak example of a snake having a gene turn on where it suddenly regrew legs. There's something out of China, but a thing to share with this, in addition to the traits reappearing and disappearing, that we are always looking for these missing links or ways to more completely build in these relationships between ancient organisms and modern day animals. And this question mark here underneath whales has already been answered since the time of this cladogram. So I just wanted to share that on the right. You can see between Egypt and Peru, we found the ancestor of whales that walked from the land into the water and still had its limbs. Okay, to keep going, this is also a vertebrate cladogram. But as you're seeing, a little bit different, still representing traits, different order or arrangement of how the different clades or groups of animals are shown, but it is still accurate. What I wanted to call out with this one though, this cladogram is showing us how information might be implied or missing. Looking over here on the side, I put this crazy little creature with a little baby on its face. And maybe you're recognizing it as a reptile, as a chameleon is what I meant. As a reptile, you might know that it's similar to crocodiles, but we don't have enough information to know, and I'll tell you, they're not the same as crocodiles. And so where reptiles would fit on this cladogram, we'd probably have to add a line or just know that they are implied. So just wanted to kind of set you up for that. You might see cladograms that seem incomplete and they are still accurate, especially if you are doing research on, 5 organisms, you maybe only want those 5 and a human on your cladogram. The top here, this slimy looking or shiny looking worm thing is an amphibian called a caecilian, but it's a legless amphibian that looks more like a worm or a snake, just showing the diversity of life. And again, the bottom here is a prehistoric shark that's no longer around. Again, with the weird diversity of characteristics that can come around, this shark had this projection, this brush-like structure on top of its head that was covered in something like teeth. The chemical composition is like teeth, which is something that worked for a while, helped that animal, and then no longer advantageous. There's something the environment changed. They are no longer around. Do you recognize in the bottom right corner, that's one of your cousins. You remember your cousin Echidna? I haven't been to Australia either, so I haven't met this echidna. That's who we're seeing in that top picture with the kangaroo looking at it a little quizzically. Because this echidna, just like the platypus down here, the other picture with the bill, they are very unusual mammals that lay eggs. So we wanted to show, here's one cladogram of a bunch of mammals in the top. This cladogram is a little harder to see, but it's showing this split of more primordial or kind of primitive, older style of mammals that gave birth with eggs. Then we have this transition, what we call crown mammals. Some of them are giving birth with eggs, some are giving live birth. And then down here at the bottom, we are placentals, we are viviparous and giving live birth. But there are mammals like this echidna, like this platypus, they have fur, they make breast milk, but they're giving birth to eggs that hatch. Very exciting. Almost done. More cladograms just to show you arthropods, the relationships, such a diversity. You may not know that lobsters and crabs are cousins of spiders and other arthropods. But it's another protein source. It's yeah, very exciting. We could talk about it more if you want. I'll stop myself. Here, this cladogram. These are different ways of showing something that's often referred to as the tree of life. So we started this presentation showing that just pictorial representation, but here, all life on Earth, bacteria, archaea, eukaryotes, have a common ancestor. And from those genetic mutations that have accumulated over billions of years, all of the life that we know is connected. This is part of why poisons and different environmental concerns can have wide-reaching effects, is because there are a lot of overlap. There's a lot of similarities between different species. I'll keep us going. Okay, here's a simple one. This is the animal kingdom. This is us and our friends. We are chordates up at the top. Underneath them, echinoderms, arthropods, nematodes, annelids or worms, mollusks, platyhelminthes or flatworms, cnidarians are things like jellyfishes and sea anemone, and then porifera, literally meaning hole carrying, hole having sponges. And this is the animal cladogram. So is this. In different order, similar arrangement, showing traits and a little bit more detail. But this is also the same information, just a little bit more. Oh, the same information, a little bit more. Is this my favorite phrase? Yes. So here showing chordates. Again, not everything with a spinal cord or nervous system has bones. So we see, again, these evolutionary advances where there will be differences in everybody following that change. So we've got cranium and we've got vertebrates, the vertebrata. Gnathostomata means a bony jaw. Once you have a bony jaw, you can bite into heavier, bigger things. Osteichthyes has a bony body. Sarcopterygians have bones in their limbs. So the fish that were able to transition from land into water. That was a big deal. Almost done. Okay, just showing part of what we're going to look at in a moment. Are sturgeon fish, or rather when you get to your bone lab, that are a little bit closer, more related to our invertebrate cousins in these cartilaginous fishes. Here again, just looking at the different groups of animals and their relationships. Here just showing one gene shared across many clades. And this is an androgen receptor, part of a steroid hormone that's involved in sexual differentiation, but just showing fish, mammals, birds, all have the same gene and the same receptor. When you get to the bone lab, Most of them are bones. There are a few red herrings, a few non-bone items. Here, coral are animals, but they're not vertebrates. So we wanted to show you this cladogram, show you that relationship, and a little close-up on the activity there goes on in coral. Beautiful coral picture if you haven't seen them in life. The diversity is pretty spectacular. Also not a bone, but might be over there, are some arthropods and crustaceans. So we've got a limpet, is really camouflaged on that rock, chitin, a barnacle. The lower left is a Pacific rock crab. The lower right is showing an ancient lobster. That's not what they look like anymore. And then maybe, do you recognize the little thing being held in the palm of a hand there? Pacific mole crab, or a sand flea, or a little sand crab, we call them. And there'll be one over there in that lab. So here, there's just an ancient mammal that's your great-great-great-great-grandpa, but 66 million years ago. The fact that so little has changed between us and our mammal ancestors, and then our reptilian cousins, all these other organisms, mean that with what you know and what you've seen, you'll be able to make some predictions. Those predictions will be on some boxes of unknown bones that I've gathered. So here on this map, that V is showing us at Vanden High. Up in the upper left, you can see an X on Clear Lake is one source of some unknown bones. And then below the V, down here, Carquinez Strait will be the source of the other unknown bones. So first, let's look at this Carquinez Strait. So again, here, us at the V, down here in Benicia is where these bones have been gathered. In between us is the Delta. You live in the largest continuous estuary, and the biggest saltwater marsh in California, the largest estuary in the United States. It is very unusual to have such a large special ecosystem. What's so special about it? You've got the Sacramento and the San Joaquin rivers emptying into that bay. That bay mixes with ocean water, and that's what we call brackish water, where it's a mix of fresh and salt water. Very unusual. I want to let you see this ecosystem so you can see what the animals in this area are eating, different types of plants. It is a salt marsh, wetlands. There are sections of oak woodland and coastal scrub, like smaller trees, grasses, herbs. Hint, cattle ranching occurred in the 1800s in this area, so cow bones might be part of what you find. To share that these estuaries and these marshes are nurseries. They're essential for the breeding of so many different organisms. Here showing an amphibian coming up for air. But we also wanted to share that what is normal and what is considered a healthy functioning ecosystem right now, that if we went back in time 50 years or 100 years, what is considered optimal would be defined differently, and what is normal will be different. So here, this is showing what does not happen anymore: a herd of elk crossing Carquinez Strait. There are no longer herd of elk in these areas. The strait itself has been dredged so that boats can get through. This is not normal anymore. Here to show this marsh, as we said, the largest continuous or interconnected estuary in the United States. Over 200 species of birds, many different reptiles and amphibians, thousands of insects. I wanted to share this one right here, the flame skimmer, this orange cousin of the dragonfly. They're related. Is an exciting one that's new to me. Some of the friends you might see in their bones, we've got osprey, mule deer, turkey vulture. The California slender salamander is endangered, but you could probably find it in your backyard. Bobcat, Anna's hummingbird. Be surprised to find some tiny little delicate bones. We've got the great blue heron, and then its lunch, a gopher. Because this is a rich, lush ecosystem with so many organisms, predators are very attracted to this, including humans. But this fisherman or fisher person holding up this big bass is a cool representation, is a representation of the concept of shifting baselines. That this today, one 20-pound fish would be an amazing catch, and for somebody working to make a living from fishing, that might be their great day of a harvest. 10 years ago, we would expect that it'd be like, ooh, I caught 2 20-pound fish. 50 years ago, it would be maybe 5 of that same kind of fish at that weight. There was a study in the Baja Peninsula with 5 generations of fishermen going back and what was considered a great catch. Many years ago, the fish were way more in number and about 2 and 3 times as big as what is considered an ideal great day of fishing for commercial fisher people now. Yeah, we can help be sustainable. That was mystery box 1 coming from Carquinez Strait. Now we're going to Clear Lake. This is another unreal, very special ecosystem. It's an inland freshwater lake. It's eutrophic, or good feeding, nutrient rich. Not like Tahoe where the water's clear because it's dead. There's not enough to feed anything. Here, just showing you some scenes from this ecosystem. Our bones are literally in this picture here, those unsullied mountains. Those are, one of them is a volcano called Mount Canocti, the one on the left. And at the base of Mount Canocti is where these bones have come from. It is the oldest lake in North America. The average lake only lasts for 10,000 years. This lake has been continuously full and able to support life for 500,000 years, which is very unusual and means that stability means a lot of richness for that ecosystem. They have many different aquatic plants, lots of introduced species too. Here's just an overview of some of the interactions that you can expect. Again, everything is food for something else in these interconnected webs. The left is showing some diatoms that if over the summer you get bored and you want to make a name for yourself, there's a lot of work to do in diatoms and soil invertebrates. Let's talk about the native species at Clear Lakes. We've got osprey, yellow-headed blackbird, pelicans, lots of birds, wood duck, Clark's grebe. The bird in the middle is your state bird. Oh, I know I can't do it. Quail. I was doing it earlier. It's the quail, California quail. You should know it. Also here, we might have mountain lions, bobcats, white-tailed deer fawn, squirrels and chipmunks. In the lower left, river otters. River otters are in this lake. And then do you recognize this big bird from Carquinez Strait? This bird gets around. Great blue heron. Okay, invasive species. Ooh, so many people are letting their turtles go. This red-eared slider turtle is competing for food with our native turtles. Brown-headed cowbird, swans. This plant is dominating the vegetation in the lake. And then the European starling is another bird that is really competing against our native birds, even though I love its rainbowness, but it's not good. Here are more ecosystem interactions. The thing I want to share with this is there are observations. I know people who've seen things that didn't make it to this data in other places. You as a scientist, as a citizen scientist, might have insights that are important to be contributed. What I had seen was an otter eat a duck, or somebody else saw it. Okay, almost done here. We wanted to share the algal blooms. You learned about harmful algal blooms earlier this year. They're a recurring problem almost every year now in the summer because of the use of fertilizers. This lake was already a rich ecosystem. It is causing algal blooms that are upon occasion killing fish, and at the mildest end causing skin irritants for humans, but solutions for you guys. Okay. In the bone lab, we can walk you through it if you're able to do this, but you'll walk through 6 different stations. Please be gentle with the bones. Please try not to spoil the surprise for others. Once you visit these stations of looking at the fish skulls, fish compared to reptile, different mammal skulls, mammals compared to birds, dropping mammal and bird bones to see the difference that they make in sound and the different appearances they have. Once you visit all these known stations, you should then be able to tackle the unknowns. Again, we got one from Carquinez, one from Clear Lake, and then unknown bones from around, mostly from our country. Please let us know if you have any questions. If anything comes up, we want to help. And yeah, hopefully we'll see you soon in the lab. And have a great summer if I don't see you.